Last week we started to talk about this great topic of statistics, and today we plan to finish it, at least most of it. So, to remind you, we started by going through a quick introduction and discussing and explaining the concept of p-values, and then most of the time last week we went through a bunch of statistical tests and tried to see when they're useful. So to remind you, we saw that the most important criterion to decide which test to use is the types of variables that we want to analyze. So we went through 2 out of 3 main cases. So we saw the numerical versus numerical case. So for example, we saw various correlation tests like Pearson's correlations and Spearman's correlation. We also saw regression, and specifically we focused on linear regression with mean squared error, what is called OLS. So we didn't speak about other types of regression, but it's something that could be useful in other scenarios, and I encourage you to look into it if the need arises. And then we also spoke about the numerical versus categorical case, and specifically we spoke quite a lot about the case where we just had a categorical variable with 2 values, so meaning we have 2 groups and we want to compare some numerical values between the 2 groups. So we saw that we could compare means using t-test or u-test, and that we could compare whole distributions using Kolmogorov-Smirnov, and then we also discussed the case of multiple options in the same category, and we spoke a little bit about the ANOVA test. So today we're going to continue with the categorical versus categorical case, which I think in some ways is the simplest case, which we have the simplest kind of test for, which are making the least assumptions. But it can also be a little bit confusing because many times when we have a categorical versus categorical case, it's not immediately obvious that this is the case that we deal with, and sometimes it can seem superficially like something else. And let's see some example and see that it can sometimes be a little bit confusing. So some examples for a categorical versus categorical association tests. So for example, we might want to test whether proteins expressed in the brain tend to have PDZ domains. So in this case, it's more obvious that we deal with 2 categorical variables. So we have one which states whether a protein is expressed or not in the brain. So we have 2 categories. And then we have a second variable which we could decide to model as a categorical variable. So we could say whether it has PDZ domains or whether it doesn't. Of course, here it's a matter of choice. We could also model it as a numerical variable and say, for example, how many PDZ domains it has. And then it would fall into a different category. Another example is 2 different chromosomes tend to express different proportions of protein encoding to microRNA genes. So in this case, I think it can be a little bit more confusing, and I think this illustrates the confusion that sometimes arises with these sorts of tests, because we have some word here, for example, the word proportion, which may seem superficially like we're dealing with something that is numeric. So we could have a higher proportion, or we could have lower proportion. But still, I argue that this kind of association test is best modeled as a categorical versus categorical variable. And the reason is, OK, so for the first variable, so for chromosomes, I think it's quite obvious that we deal with something that is categorical. So you have different chromosomes. It's not something numeric. Now, what about the proportion of protein coding microRNA genes? So I think the hint here is what kind of analysis we're going to perform when we try to carry out this sort of analysis. So the thing we're going to do essentially is just to count genes. So we're going to count protein coding genes and microRNA genes. So each gene can be either protein coding or microRNA. And in the end, we might get some table, which is called a contingency table. I'm going to discuss it in a few slides, which says for each combination of a chromosome and type of gene, which could be either protein coding or microRNAs, how many of these combinations we have. And since we're in the business of counting objects, that's typically a big clue that we're dealing with two categorical variables, okay? So we look at two categories, and then we count how many entities we have of each combination of these categories. Another example is our targets of specific microRNAs more abundant on specific chromosomes. Again, we have the word abundant, which may seem a bit confusing, but again, we're in the business of counting here. So we look here the same way. We look at the genes, and they can be either targets of those microRNAs, or they can be non-targets. And then they can belong to any of a given set of chromosomes. So again, two categorical variables that we deal with. And finally, a final example, for example, if we want to see whether some variants are associated with specific phenotypes. So again, we can think about it as a 2-by-2 kind of combination. So for each variant that we look at, we can look at patients or individuals that have this variant and individuals that don't have this variant, and then individuals that have the phenotype and individuals that don't have the phenotype. And again, we're in the business of counting individuals that might have this combination. Any questions? Yeah.
The homework exercise would have the 4 variables to make a contingency table. Would it be possible to do that?
Sorry, the homework exercise?
Yeah, the B, it was pathogenic. What was it exactly?
Pathogenic or benign.
No, the B was
Portion of
pathogenic.
pathogenic in the
Yeah, percentage. And I was wondering if it's also possible to do a contingency table to get the 4 variables accounting for this question. Was that possible?
I don't exactly remember already. We can speak about the homework maybe later, but I think there it wasn't the case. But yeah, we can speak about it. So again, if you have one variable which is categorical, but the second one is numeric, then that's not the case. So that only works for two categorical variables, when you actually count combinations. So let's begin with the simplest case of a 2-by-2 scenario, okay, which is actually quite prominent. And as we will later see, we can also reduce many of the problems to this case. So as a simple example, let's consider a simple test that we may want to carry out to see whether there is some correlation between smoking to lung cancer. Okay, and then we end up with a 2-by-2 contingency table that would tell us for each combination how many individuals we have. So individuals can be either smokers or non-smokers, and they can either have lung cancer or they can be healthy. And then we also have the total numbers, which is not really part of the table. So it is only a 2-by-2 table. And when we have that case, we can use a simple test, which is called Fisher's exact, which you already saw and worked with in one of the previous exercises. So the input Fisher's exact test would typically be just a 2-by-2 contingency table, like you saw before. The purpose of the test is to test whether the rows and columns of the table are associated with each other. And then the null hypothesis would be what is called a hypergeometric distribution, meaning there is no association. So a hypergeometric distribution is what we get when you have two binomial distributions, so for the rows and columns, which are independent. And then their combination would be that distribution. And the nice thing about it is that you don't need any additional assumptions. Okay, so it's exactly modeled the right way. So unlike many of the parametric tests that we saw, we don't assume any approximation to normal distributions or stuff like that. Yeah, question?
Can you just confirm again the hypergeometric distribution, just two binomial distributions that are independent from another?
Yeah, exactly. So if you have one variable which is binomially distributed and another one which is binomially distributed, and they're independent, then their joint distribution, so which combination you get, will be a hypergeometric distribution. Yeah. So in some way, it's more like the non-parametric tests that we saw that don't make any assumptions about more specific distributions. Now, it's a very, very powerful test, and it gives, as the name suggests, it gives exact results. But the limitation is that it's not practical to do it for large tables or large sample sizes. So typically, it will only work for 2x2 tables. And the reason is, I think you saw how it works in the video you got to watch in the exercise, where you had the tea lady experiment, if you remember. And then the idea was to take the kind of observation that you get, and then to consider all the possible tables that would be even more extreme than the kind of table that you get. And the nice thing about it is that you have what is called only one degree of freedom. So as soon as you know one of the combinations in the table and you know the total numbers, then this uniquely determines all the other slots in the table. But that's only the case in a 2x2 table. If you have a 2x3 or a 3x3, then you have many more degrees of freedom, and it really grows exponentially. So the more slots you have in a table, the more combinations you have, and then it becomes impractical to actually calculate it. And you saw the kind of implementation in SciPy. Now, it's important to state that like virtually all other tests, we also, of course, have the assumption that we must have the samples to be IID, so they have to be independent. And very importantly, again, like all other tests that involve categorical variables, we need that the categories will be mutually exclusive, okay? So if they're not, we need to define them in such a way that they will be mutually exclusive. We cannot have any overlap in the definitions of the categories.
For numerical tests, that doesn't have to be the case, that they're exclusive.
So in numeric variable, I don't even know what it means to be exclusive, because it's just a number. So
For example, the one that you had in the exercise with the percentage of the pathogenic variants and all variants.
the percentage of pathogenic variants.
With variants, with all variants, and then the percentage of them which are pathogenic. They're not exclusive, I would say. It's like one variable is like
Yeah, but
the
then
other one.
then you're not talking about the numerical variable, you're speaking about a categorical variable. So of course, when you have a categorical versus numeric, like for example a t-test or u-test, then again it has to be two different groups. You cannot have this overlap. Okay? Okay. So now a Fisher's exact test will actually give you the significance of the association, but it won't give you the strength of the association. So if you want to actually measure how strong the association seems to be, you have a few options. The simplest kind of metric that we could use to actually measure the strength of association between two categorical variables, that each of which has two options, is called the risk ratio, or RR for short. And that's defined as followed. So very often when we speak about a 2x2 case, we can call one of the variables, we can consider it to be some kind of risk factor, and the second one we can consider it to be some kind of outcome. So for example, it could be a disease. Okay, so traditionally when people define it, they speak about disease and some exposure risk factor, but of course it can be any kind of a variable that we look at. But if we speak about diseases and exposure, then the way we would define it is the probability of having the disease given that you are exposed to the risk factor divided by the probability that you have the disease given that you are not exposed to the risk factor. So it's quite intuitive. What it essentially means is how much more likely you are to have the disease given that you are exposed than if you were not exposed. So for example, in the case of lung cancer and smoking, a risk ratio of 3 would say that if you smoke, you're 3 times more likely to have cancer, which is very intuitive. So it's a very nice measure, very intuitive. But the problem is that it's sometimes hard to design a proper cohort study that can give a reliable risk ratio. And the reason is that many times we don't actually collect the data this way. So if we collected exposed individuals and unexposed individuals, and then just saw what they end up being, if they end up having the disease or ending up not having the disease, then there is no problem. We can actually calculate the risk ratio. But many times the disease is very rare, and designing this kind of experiment just won't work because we won't have enough individuals with the disease. So for example, if we deal with a disease that is 1 to 1,000, then if we just collect 500 individuals, then we won't even have a single patient with the disease, and we won't have any statistical power to do anything about it. So many times what people actually do is actually going the other way around and collecting patients that have the disease and collecting patients that don't have the disease, and then for them checking if they have the risk factor or not. So that, for example, if you look at this example, for example, you can see that this seems to be what's going on here. So you can see that we have exactly 400 lung cancer patients, and we have exactly 600 healthy patients. Okay, so this suggests that this is actually the way they were collected. We didn't just collect a random sample. We explicitly built this kind of case control study where we took cases and we took controls, and then we asked them whether they smoke or not. So we did it the other way around. By the way, these numbers are completely arbitrary, so don't take them seriously. But anyway, yeah, if we have that kind of data, we cannot really calculate the risk ratio because we don't know these conditional probabilities. And then we need to use some other metric. For example, a very popular choice would be the odds ratio.
Okay, why don't you have this data? I mean, you have this equilibrium.
Not exactly. So I wanted probability of someone having the disease given that they are exposed. So in our case,
they're not smoking.
so given that they smoke, for example. So given that the person smokes, we want the probability that they have lung cancer, okay? But that's not actually the way we collect the data. We didn't take people that smoke and then checked, okay, how many of them have lung cancer, how many of them don't have lung cancer. That's not what we did. We took people with lung cancer and we took healthy individuals. So we know the probability of someone being a smoker given that they have lung cancer, but that's not the same as the probability of having a lung cancer given that you are a smoker, okay? So we need to remember that conditional probabilities are not symmetric. Okay, that's not the same.
Why can't you just take 400 lung cancer patients and ask whether they smoke or not?
No, we do it. We collect, that's exactly what we do. We collect 400 lung cancer patients, and then we check how many of them smoke and don't smoke.
That's exactly what we...
But here we have it the other way around. So we don't want the probability of smoking given that you have lung cancer. We want the probability of having lung cancer given that you smoke. Okay, it's just unfortunate law of probability that it doesn't go the other way around. Yeah.
Would you right to say that this is sort of like equivalent to the strength of R, let's say, in Pearson when we talk about correlation?
Sorry, say it again.
Equivalent to this, like to R in Pearson correlation, when we're talking about the strength of the correlation between two variables?
Yeah, so yeah, exactly. That's kind of a measure. So in Pearson's correlation, we saw that the R coefficient is a good measure for the strength of the association, which is very distinct from the p-value. So here again, so we have the p-value that we could calculate using Fisher's exact test, but now we also want some kind of measure for how strong the association is. Okay. So because of this asymmetry, which prevents us from calculating the risk ratio, in many cases, there is a different measure which we could use in case control study design, which is called the odds ratio. And that's why the reason it's more common than the risk ratio, even though it's less intuitive. So the definition of the odds ratio is more complex. So we have this somewhat non-intuitive entity, which is called the odds, so we take the probability of being exposed given that you have the disease divided by the probability of not being exposed given that you have the disease. All of that divided by the ratio of being exposed given that you're healthy or not exposed given that you're healthy. So the important thing to note here is that all of the conditional probabilities here are actually reversed. So here we condition on having the disease or being healthy, and not on being exposed or not exposed to the risk factor. And that's why we can actually calculate it even if we collect it the other way. Now, the nice thing about this metric is that it's distinct from the risk ratio. It doesn't give the same number. But if we deal with a rare disease that doesn't happen a lot in the population, then we can actually prove that this is a quite good approximation of the risk ratio. And that's exactly the kind of cases in which we are forced to use the odds ratio instead. So if we have a disease which is not rare, we can just collect ordinary data and calculate the risk ratio. And if we have a disease that is rare, we can calculate the odds ratio instead, and that would be a good approximation to the risk ratio. And that's actually very easy to calculate it given any 2x2 contingency table. You can actually take the counts here and just divide them according to this formula. And it's even fully symmetric, so it doesn't matter what you define to be the risk factor and what you define to be the disease. So if you transpose it and turn the columns into rows and the rows into columns, it wouldn't matter to the odds ratio. So it's fully symmetric and actually agnostic to transposing the rows and columns. Any questions? OK. So we spoke about the 2x2 case, but what happens if we have larger tables? So we have more options. So for example, we may, instead of just defining smoking and cancer as two options, we may want to consider more options. So for example, in smoking, we can define heavy smokers, light smokers, and non-smokers. And we may want to consider various types of cancer. And then we wouldn't be able to use the Fisher's exact test like we saw. But luckily, we have a different test, which is called the Pearson's chi-squared test of independence. The input of the chi-squared test is any size of contingency table. And like the Fisher's exact, it will test whether the rows and columns are associated. Again, under the null hypothesis that rows and columns are independent. But unlike Fisher's exact test, this one is not exact. So it does make additional assumptions. And like most parametric tests, you will need to make sure that the assumptions hold. So in this case, the assumption is that the expected cell values can be approximated by normal distribution. I will explain in a minute what exactly it means. Yeah, and another important limitation is that like all omnibus tests that we saw, so it's very similar to a ANOVA test, where we could determine that there was some association, but we couldn't tell which are the two groups that have different means. So here as well, you will get a significant result, meaning that there is some association between the two categories, but you will not know exactly which categories are the significant ones. You just know that overall, there seems to be an effect. So how does this test work? So it's actually quite simple. So what it basically does is just comparing the expected values of cells under the null hypothesis of independence to what we actually observe in practice. So this is called the OIJ, is the values within the table that we actually observe. So I is the index of the row, J is the index of the column. And then we compare this to EIJ, which is the expected values, which you can calculate very easily using the formula of taking the total of the row, multiply it by the total of the column, and dividing it by the grand total. Now, like I said, the assumption of the chi-squared test is that the binomial frequencies of cells can be approximated by normal distribution. Okay, so it means that the expected values, not the observed values, must not be too small. And a common rule of thumb that people use is requiring that we have it larger than 5 for at least 80% of the cells, and larger than 1 for all the cells. And again, I remind you that we speak about the expected values. So don't look at the observed values. You need to actually calculate the expected values. We're going to see how you do that. And then look at them. So even if you have some combinations which don't occur at all, it can still be a valid thing, because maybe you don't observe them at all, but if it were independent, you would still expect to see some of them, and then it's totally fine, and there is no problem with that. And of course, like always, you need this criterion of being mutually exclusive, and of course, you need the samples to be independent. And let's see a quick demonstration of how this works. OK, so I opened the same notebook from last week in the section of contingency tables. And I just hard-coded here the same table that I gave as the example of the lung cancer in healthy versus smokers or non-smokers. And let's see how we can use Fisher's exact and chi-squared test on this data. So we import both from SciPy stats, both tests. Now using the Fisher's exact, like you saw in the exercise, is super simple. We just feed in the table, and we get the odds ratio and the p-value. And you can see that we get an odds ratio of 4.12 and a p-value on the order of 10 to the minus of 26. So it seems that smoking, according to this fake data, is very strongly correlated with lung cancer. So it's very significant and very strong. So roughly, if we see the odds ratio as a close approximation of the risk ratio, we could say that if you smoke, you will be 4 times more likely to have lung cancer. OK, just to show you that SciPy's implementation knows how to calculate the odds ratio correctly, and that the formula that I gave you actually works, you can see here we can actually manually calculate the odds ratio by just dividing all of the 4 values within the table, and you see that we actually get the same result. Okay. Now, if we want to use the chi-squared test, again, we'll just call the function and give it the contingency table as input. And then we're going to get 4 parameters. So the first will be the chi2 statistic, which typically we don't care about. Then the p-value, degrees of freedom, again, not that important. And the expected data, which is important. So you can see that the p-value is quite similar. So here we, as expected, it doesn't matter too much which test we use. So the chi-square is indeed a good approximation of the exact calculation. It's slightly more conservative, so just on the order of 10 to the minus 25. Not that we care too much in this case. Now, by the expected table, you can see that we can actually print it to the screen, and you can see that this is what we would expect to get if smoking was independent of cancer, and that's what we actually got. So you can see the difference. You can see that in reality, we have more smokers with lung cancer than expected, and likewise for the other combinations. And you can also see that we definitely have no problem with the criteria we defined. So you can see that the expected table, all of the values are very high. So the kind of rule of thumb that we discussed is fully met here, and it's indeed valid to use the chi-square test in this case. OK, let's see a second example that here we will be forced to use the chi-square test. So I wrote a piece of code here that uses the gene code data set to count the number of genes that are defined as protein coding or microRNA per chromosome. I'm not going to go over the code. We've seen like 100 examples of this sort already. So you can go over it later if you want. But the end result is we get this nice table, and now we want to see whether there is some association between the kind of genes we have, either protein coding or microRNA, to different chromosomes. So again, we can simply use the same chi-square test. And so again, we get a p-value and expected table. And now we actually wrote this code that will check if we have those criteria met. So you can see we compared the expected data at 5 and calculate the mean to get the fraction of the entries which are larger than 5, and again, the fraction of the entries which are larger than 1. And you can see that 98% of the values are above 5, and 100% are above 1. Again, I remind you, it's the expected values, not the observed values. So we actually have the rule of thumb, which we spoke about met here, and it's valid to use the chi-square test. And we get a p-value which is very significant. So we can conclude that different chromosomes do have tendency towards different gene types, but we don't know which are the chromosomes with significantly unexpected tendency. So that's the limitation of the chi-square test, that we don't end up getting a very concrete answer. And also, it's very important not to confuse significance with strength or effect size. And that's a very, very common mistake. So you will very often see people making some claims like, OK, we got a very significant p-values. So we see a very, very strong associations between different chromosomes to different kinds of genes. And that's actually a wrong inference. So just because it's very significant doesn't mean it's very strong. So we can later look at it and actually see that we see only very mild differences between chromosomes. So it could be a very small effect, just because we have a lot of data. And that's the only reason why we got significant results. So if we want to judge the effect size, we have to do it independently from judging the significance. Questions? Yeah.
So in the Fisher's test where you do get a ratio, a relative risk, so how do you get the confidence interval in that ratio?
The confidence interval. We're going to speak about it later. OK. Any more questions? OK. So that pretty much covers the basic statistical test, so the larger part of our discussion. And now I want to go over some. Yeah.
So you mentioned that you cannot really make an inference of which of the 20,
Yeah.
but let's say you really want to do it to
Yeah.
understand this. So
How you do that? Yeah. So OK. So I forgot to mention that. So if you want to after carrying out a chi-square test to determine which groups are actually different, you need to use what is called post hoc tests, which I won't speak about because it's a more complicated topic, but it's something you can read about in textbooks. The kind of option that we're going to see later is doing some kind of an all-by-all comparison and then using multiple hypothesis testing correction, which will allow you to actually figure out which groups are different from whom. So I'm going to speak about it later, and we're going to see exactly with this example, we're going to see how you can figure it out. OK, so some caveats which are related to statistics, which I thought is very important to mention. First, I think this data set, again, will look familiar to you. You worked, it was some kind of an unseen data set that you got to work on in one of the exercises. And I think you got the impression that this was a bit confusing, because in the exercise, you had to first calculate a bunch of statistics, and then, without even seeing the data, you had to guess whether they are similar or not. And you can see that in reality, they look very, very different, these four data sets, despite the fact that they all have identical statistical properties. So they all have the same mean of x. They all have the same variance of x, the same mean of y, and the variance of y. They have the exact same correlation coefficient and correlation p-value between x and y, and the exact same regression line, as you can see here. OK, so from if you are a blind statistician, you may think that those data sets are exactly the same. But if we actually look at them, we understand that they look very different. And that's actually kind of an artificial data set that was specifically constructed for this educational purpose in 1973 by the English statistician Anscombe, which wanted to demonstrate a few important things that he thought that statisticians at this time didn't appreciate enough. So the first one was the importance of actually looking at the data, which is very often neglected because people have the sense that statistics is very accurate and very objective, and graphs are very subjective and inaccurate. And the second thing he wanted to demonstrate is the importance of treating outliers properly. So you can see that in those two last examples here, in the third and fourth of these 4 data sets, we have pretty much the whole signal just driven by outliers. So in this case, we have perfect correlation other than this outlier. In this case, we don't have anything. And the only reason that we think we have correlation is, again, because of this outlier. So I think it's a very nice demonstration for some lesson that people tend to forget, that we should be skeptical of the notion that numerical calculations are exact, but graphs are rough. So I think it's very misleading. Sorry, and the right way to look at it is to understand that both graphs and statistics can be very, very misleading. And it's very, very recommended to actually combine the two of them together. So every time that you actually make an analysis, I think it's very important to both look at the data and to analyze it. So we saw many examples that graphics can be misleading. So we can see all kinds of differences, which might not be statistically significant. But now we see that it can also go the other way around. So it's important to do both, I think. And then you're less likely to get things wrong. And again, it's also important to be very skeptic about everything that you get through the results, and actually really trying to understand where the signal comes from and what it actually means for you and what are the right conclusions. So actually thinking about the results you get and not just jumping directly to conclusions. So if you get a significant result, thinking that it automatically means that whatever you hypothesized must have been true, I think again is very important. And yeah, that reminds the kind of phrase that I wanted to remind you. So remember your solemn duty as a researcher to be skeptic and not to abandon an hypothesis too quickly. Another important caveat that I think is important to mention, which is related to the previous example, is the way that we need to deal with outliers. And we saw many examples. So we saw in this Anscombe's quartet that outliers can really mess up the results. And we also saw some more concrete examples. So we saw with viruses, if you remember the first example where we ran Pearson's correlation, and we saw that it was really, really dominated by just 2 viruses, which were outliers. So actually, most of the statistical tests that we learned and most of the parameters, so if you calculate the mean, standard deviations, stuff like that, it will be very, very sensitive to outliers. So again, most of the tests that we looked at were very, very sensitive. Some are more robust. So if you use the median instead of the mean, or if you use Spearman's correlation instead of Pearson's correlation, or a U-test instead of a t-test, then you're on a much more stable ground with respect to outliers. And yeah, and actually deciding what to do with them is not always trivial. So I think there isn't like a manual or a recipe that I can give you, so you need to always follow these 3 rules. And that's the way to deal with outliers. I think it really requires knowledge about the kind of domain that you're analyzing, and it requires a lot of judgment. There are, however, a few rules of thumb which I think most of the time should be applied. So for example, if it's obvious that your outlier is actually a wrong measurement, so it's something that was incorrectly entered or something that was a mistake, then you should drop it, of course. If the outlier creates a significant association that otherwise wouldn't exist, then I think it would be dishonest to report significance in this case, because clearly everything is just dominated by a single outlier. Now, if you do choose to drop data, then of course you must report that. And in any case that the presence of specific outliers really significantly changes your analysis, it should be noted. Okay, so if you see that your analysis is really dependent on 1 or 2 outliers, then it would be wrong just to report one analysis that either considers them or not, and you should really note that they make a difference. And of course, it's also a very important red flag that you should seriously consider to use a more robust test like we saw before. That's the figure I really like, which teaches the important lesson of being wary of very small effect sizes. So it says, I don't trust linear regressions when it's harder to guess the direction of the correlation from the scatter plot, like you see here, than to find new constellations on it. And I really think it's spot on. So like we said, even if it's significant because we have many, many points, I think you would agree that it really doesn't mean much. And again, this resonates with the lesson I spoke about of distinguishing between significance and effect size. Another super important caveat, which seems obvious at first, but I think in real life it can be very, very confusing, is this notion of sampling bias. So what it essentially means is if you collect data on one population, then you cannot come to conclusions about a different population. So if you do statistical analysis on apples, it won't tell you anything about oranges. So in principle, it seems quite obvious and easy to follow. But I think you can see that in many real-life situations, it's more difficult. So let's, for example, consider this story. So here we have Dr. Sloppy, who wanted to see if there existed a correlation between food poisoning and ankle sprains. So what he did is surveyed the patients at the St. Pearson's Hospital and counted how many have none, either, or both conditions. And he found significant negative correlation between the two of them. So what it means is patients who had one were less likely to have the other. So if you had food poisoning, you're less likely to have ankle sprains. And Dr. Sloppy concluded that pathogenic food bacteria are protective from orthopedic trauma. Now, what do you think might be wrong here? Except, of course, from the last, which is the last conclusion, which is obviously very arbitrary. But even if Dr. Sloppy had just settled on a more, on a more basic conclusion, you would just say that there is negative correlation between food poisoning and ankle sprains. What would you think about it?
Selection bias.
Okay, yeah. So the title is kind of a giveaway. But why selection bias?
It's sampling a specific population, people that are already in the hospital for a specific reason.
Right. Okay. So you get the problem. So people that are hospitalized are not a good representation of the overall population. And by the way, can you think of a reason why you would have negative correlation between those conditions in hospitals?
Just lay on the bed.
So you said right. Okay, they came to the hospital for a reason. Okay, so food poisoning and ankle sprains can be two of the most common reasons why people come to the hospital. So they are there for a reason. So if they don't have one of them, it's very likely that they have the other one. And if they have one of them, it already explains away why they're in the hospital, and most probably they won't have the other one. By the way, it's a very, very common phenomenon that epidemiologists observed, that within hospitals you tend to have very strong negative correlations between conditions, exactly for this reason, even for conditions which in real life have nothing to do with one another. And when put out of context, this can really be very confusing. So actually, so many statisticians have struggled for quite a long time to understand this phenomena. And it actually wasn't at all trivial for many researchers why this happens. Just to illustrate that in real life, when you come across sampling bias, it can very often be very subtle and hard to notice, but very significant when this actually influences the results of your analysis.
But it would be true to say that within the hospital population, there is a negative population.
Exactly, yeah. Yeah. So it is true that within hospitals we have negative correlation. It just wouldn't be right to generalize it.
This specific hospital.
Yeah. And yeah, remember. Okay, another important caveat is, again, I think all of you are familiar with this notion that correlation does not imply causation. So we have that correlation or association is fully symmetric between two variables. But it doesn't necessarily mean that either of them causally influences the other. So very often we have the problem of confounding. So we could have a third variable z, which affects both x and y, and that explains why they are correlated. OK, and actually inferring causality is very difficult without experiments. Because if we just observe data, it's very difficult to know what is the reason behind it. So the classic way to actually interrogate that is to run experiments. So we need to actually try to change one variable and then see if that actually influences the other one. And the gold standard when it comes to those kind of experiments is, of course, what is called a randomized controlled trial, or RCT for short, which is commonly used in medical practice. So if you want to see that a drug actually helps for some condition, we would have to take samples and then to randomly split them into two groups, one that is treated by the drug and one that is not treated by the drug. And then we would actually have to see whether it makes a difference after we have actually interfered and ran these experiments. So then we would actually be able to come up with causal conclusions. Now, I'm not trying to say that associations are completely useless. So they are very important, and they will give you strong evidence. And many times we don't have the luxury of running experiments. So many times it's impractical or unethical to run experiments, and we have to use other means. So I think the right way to look at it is seeing associations as one layer of evidence. And if you have many independent evidence that can support some conclusion, then you are more likely to accept that. But you should avoid jumping straight ahead to conclusions. And when you see association, thinking directly, OK, that must mean that we have some causal relationship. A final thing I wanted to mention in this context is the issue of data normalization. And I think it will be easier to illustrate that through an example. So let's say that we want to check whether brain-expressed genes are enriched with genetic variants, so whether they have more variants than average. Then we would likely encounter a problem that it could very well be that brain-expressed genes could be on average longer or shorter than regular genes. And if that's the case, we would obviously see more genetic variants because we expect for longer genes to just have more opportunity to accumulate variants. And then we would have this confounding, and we wouldn't know whether brain-expressed genes tend to have more variants because of some important biological or evolutionary reasons, or whether just as a side effect of this observation. So there are many ways to try to overcome that. One simple way is just to do what is called a normalization and try to normalize the number of variants by the gene length. So instead of looking at the number of variants, we would look at the frequency of variants per nucleotide, say. But then again, it could be a problem because when we normalize some variable x by some variable y, then we typically assume that they should have linear relationship. Okay, and it could, in many cases, that's not necessarily the case. So in this case, it seems reasonable to assume that it should be linear. So if you have a gene that is twice as long as another gene, it's reasonable to maybe expect that it will have, on average, twice as many variants. But in many cases, it could be dependent, but not exactly linearly. So maybe it's only sublinear with respect to gene length.
I don't think it's a reasonable.
You don't think it's a reasonable assumption?
No.
Why not?
Because, for example, some genes may have many exons in which variants are very rare, let's say, and the number of introns is or something like that.
Yeah.
So there are biases in this length that are not equal, that are very different, let's say, annotation-wise, and stuff like that. Like 3' UTR, which is huge, take 30% or 50% of the gene length in one case, and it can accumulate as many variants as you
Yeah.
want, but the other will, and so on.
Yeah, it makes sense. Okay, so I think it illustrates the example, the idea I was trying to make, that normalizing can be very tricky because, again, like Michal says, you don't always know whether it's a reasonable assumption to expect that they would behave, really tightly, closely together. And then many times you would have to settle for another option. So for example, in this case, we might be better off to just compare genes of comparable lengths of say, brain-expressed genes to genes that are not expressed on the brain. So that's one option, just to look at groups which appear to be similar. Another way to do that is through multivariate analysis. So we could treat the gene length as some kind of covariate and try to control for it. Now, I'm not going to speak about that today, but it's actually going to show up next week. So next week, I'm going to speak about how we can do that. OK, so I threw in a lot of caveats and telling you how statistics can be useless, and you should be very careful and not to jump to conclusions. I don't want you to get the impression that I think statistics is actually useless and that you shouldn't use it and come to conclusions from it. Just that we need to be careful when dealing with it. And yeah, and when we do a careful, proper statistical analysis, it can really teach us a lot. And yeah, so even if we don't actually prove causal connections, it's still an important layer in establishing the kind of claims that we want to make.